My name is Iain Hagan, and I want to tell you about an exciting opportunity for doing research in my group here in the CRUK Manchester Institute. We study the control of cell division and how these controls change when the environmental context of the cell, outside and inside the cell, change. So progression through the cell division cycle essentially is driven by the sequential activation of a series of cell division kinases called CDK cyclin kinases. Now the CDK cyclin complex is composed of a catalytic CDK subunit and a cyclin subunit, and it is the cyclin subunit accumulates as the cells go through the cycle until the action of the complex is complete, whereupon the cyclin subunit is destroyed. And in many respects, it is this sequential accumulation and loss of CDK cyclin activities that drives the progression through the cell division cycle. However, there are finer controls, and it's the finer controls that determine exactly when each accumulated complex will be activated. So on the one level, there are small molecules, small proteins that inhibit the activity of these CDK cyclin complexes, and these proteins can be stimulated by growth factors or DNA damage responses. And then on the other hand, there is a post-translational modification of the CDK cyclins to inhibit the activity of the kinase directly. So the post-translational modification of CDK cyclin activity to regulate it is best understood in the case of CDK1 cyclin B. Now CDK1 cyclin B presides over the decision to physically separate the genome in mitosis. And CDK1 activity is inhibited by Wee1 kinase. Now Wee1 kinase phosphorylates the active ATP binding pocket of CDK1 when it's in complex with cyclin B, and this stops the CDK1 from being active. It means the cell can accumulate a large stockpile of inactive CDK1 cyclin B until the time for division is right, when CDC25 phosphatase rips off this phosphate and you now release a large burst of active CDK1 cyclin B. Now when DNA is damaged or DNA replication is incomplete, there are checkpoint pathways that boost the activity of Wee1 and repress CDC25 activity. Consequently, the cell will not undergo or even attempt cell division until the DNA damage has been repaired. Now these controls that act over the timing of genome segregation through Wee1 control of CDK1 cyclin B are particularly heavily used in tumour cells, because tumour cells accumulate more damage and have less controlled DNA replication than the neighbouring normal tissues. Consequently, they have a far greater reliance on Wee1 function than the neighbouring normal tissues. Now for this reason, inhibitors of Wee1 have been developed, and one in particular, AZD1775, is performing extremely well in preclinical trials and is therefore being progressed into clinical trials. So there are three members of the Wee1 kinase family in humans. There's Wee1 itself, MYT1 kinase, and Wee2. Wee2 is a meiosis-specific version that is only used in the gonads, whereas both MYT1 and Wee1 are expressed in somatic tissues. So the inhibitor that is doing so well in preclinical and being advanced into clinical studies, AZ1775, works 100-fold more effectively against Wee1 than MYT1. And consequently, we don't really know whether inhibition of MYT1 would be of any benefit for therapeutic exploitation. So the project that we would now like to recruit to is to conduct a very basic characterization of MYT1 kinase function. We want to ask some very basic questions about what MYT1 does. You know, is MYT1 contributing to the inhibition of CDK1 cyclin in normal or perturbed cell cycles? Is there a tissue-specific variation in the reliance upon either Wee1 or MYT1? Those core questions, we really don't know the answers to them. And so the project would exploit all the latest up-to-date technologies to interrogate MYT1 function. Now, we would like to use CRISPR to edit the genome to create cell lines in which MYT1 kinase can be inhibited by ATP analogues. In the same kind of approach, we would create cell lines where the sequences encoding MYT1 are fused to sequences encoding fluorescent proteins or epitope tags, and that would allow us to do the biochemistry to monitor the activity of MYT1 and the distribution of it. We can follow by microscopy as cells go through normal and perturbed cell division cycles. Similarly, polyclonal antibodies could be used in similar studies, but polyclonal antibodies would also be extremely useful to interrogate the vast array of tissue samples we have in the biobank here in the Manchester Research Centre, where we can address the question of whether WEE1 or MYT1 kinases expression is altered in a tumour context. And then we will build a research programme on these findings. So this work will be conducted within a dynamic and vibrant group. We are working on the control of cell division. Half the group works on exploitation of yeast as a model system to understand the core principles of cell cycle control, whilst the other half is using human cell lines to look at cell cycle control and protein phosphatase function. So our group works within the CRUK Manchester Institute, which is a highly dynamic international cancer research institute with groups working in a range of different areas of cancer research. Some of these groups, our neighbours, are working on the interrogation of WEE1 function in preclinical models and conducting clinical trials in the neighbouring Christie Hospital. We therefore believe this is a very exciting time, but also a very exciting place in which we can conduct a long overdue interrogation of MYT1 function. And I firmly believe that we're going to find out some really exciting biology that's going to not only change the way we understand the cell cycle, but point ways forward for some really exciting approaches to cancer therapy.